Hey Guido Fam Bam, it's Mrs. Guido. I'm going to go over the phases of meiosis with you guys to make sure that you are prepared for your quiz. Your quiz is going to be tomorrow, which is Friday. You do have a worksheet that's posted in Canvas as well that's due tonight, just a refresher on meiosis. Hopefully that activity, the video activity, helped you guys go through your notes and try to remember all the little nitty-gritty details of meiosis. But there were a few videos that I came across that people had slightly incorrect information. So I wanted to make sure that I cleared the air and made sure that everybody's on the same page. So when we're talking about meiosis, remember, meiosis is the process of making gametes, and gametes are the sex cells. So in males, we will be making sperm, and in females, we'll be making the eggs. The sperm will end up with 4 total sperm, and eggs will end up with 1 egg, and then 3, they're called polar, bodies. So we end up with 1 egg, and then 3 polar bodies. The 3 polar bodies, so like the egg is like super big, it's hoarding most of the cytoplasm, and then we have these like little baby cells. So we do end up making 4 cells, so only one of them becomes the egg in females. Where in males, all 4 of them become little spermies. Each of these cells end up with 20, for humans, 23 chromosomes. So all of these would have 23 chromosomes in them. And again, that's important because when the sperm fertilizes the egg, now our zygote will end up with 46, and that is the correct number of chromosomes for humans. Okay, so before meiosis begins, we do have interphase, G1, S, and G2. S phase is where the DNA is making a copy of itself. So if this is our like little strand of DNA, the DNA is going to go through replication, DNA replication, which we had a unit on that, and it makes a copy. So that's where we end up with that like chromosome X looking thing. Remember, the centromere holds the 2 sister chromatids. So this is 1, 2. And remember that the sister chromatids are identical to each other. So each sister chromatid is identical. And we have 46 of those making copies. So that's 1, and then we would have like another one, and it's going to make a copy held together. And these 2 sister chromatids in this blue one are identical to each other. And the 2 for the purple color, maroon, I don't know, whatever the color that is, they're identical to each other. But the maroon is not the same as the blue, right? They're coding for different genes on these different sets. Okay, so after we get through interphase, then we enter into meiosis. Meiosis is broken down into 2 phases. We have meiosis 1, and then we have meiosis 2. So meiosis 1, we'll just say the whole thing's meiosis, right? So meiosis, meiosis 1, hold up, there we go. Oh, snap. Okay, we start off with prophase 1. In prophase 1, we have our cell, right? And I'll go ahead and draw it in. We have our centrioles. Our centrioles begin to separate from each other, so they're going to be going to opposite ends of the cell. So I'm going to move that guy over here. We have spindle fibers that begin to form. Our spaghetti-like DNA begins to coil up into a chromosome state. So we can now see our DNA. I'm actually going to make those into different colors, so give me a second. Hold up. We'll do one purple. Cool. And okay, so again, centrioles separate, spindle fibers form, DNA condenses into chromosomes, the nuclear envelope breaks down. So those are the four things that are happening in meiosis that are also happening. Sorry, those are the four things that happen in mitosis that also happen in meiosis. So centrioles separate, spindle fibers form, DNA condenses, nuclear envelope breaks down. There's two additional things that happen in meiosis, and that's where so the additional two things are homologous chromosomes pair up, and that forms a tetrad. Tetra, remember, means like four, so we would have like that one, and we could say maybe that one and the yellow one are homologous to each other, so they're pairing up. And then our, what is it, blue and pink, and blue, they're homologous to each other, so then they're pairing up. So then I'd have to show that over here. So they're moving around, and then we would have those pairs pairing up. Cool. So that is one of the steps. And then the next step that happens in prophase that is different than mitosis is crossing over. So it's called synapsis, and think of it like a snapping, like we're snapping a piece of the leg and it's flip-flopping with the homologous chromosome. Remember that this technically happens in more than one spot, but just for simplicity, we're going to just represent it as one. So we'll show this one. And grab that yellow. So there's one little leg. So we have crossing over between those two, and then crossing over is also happening between the other two. Oops, that's the wrong blue, right? Okay. Cool. All right. So these homologous chromosomes pairing up, forming a tetrad, and crossing over happening, those are the two new additional things that happen in meiosis. Now, these two things are insanely important because this leads to genetic variation, right? So now our sister chromatids, this one and this one, they are not identical anymore. They are genetically different because I have, if we were to highlight the one side, highlight it in yellow, no, we'll say green. So this side is all purple, right, or maroon, and then the other side, it has maroon and a little bit of yellow in it. So they're not identical to each other anymore. Cool. And that leads to genetic variation, which genetic variation is a good thing. We want genetic variation. We want genetic variation because if there's some type of environmental change, the species as a whole might not be suited for this new environment. They might not be quote unquote fit for this new environment. But if we have genetic variation, then there could be some organisms within that species that have beneficial traits. So they might be able to, so a select few might be able to survive. And as a species, as a whole, they could survive, right? So the species could survive, even though a lot of the organisms might not make it, they might not thrive in this new type of environment. There might be some individuals that have some beneficial traits and so that they can survive. Okay, so after we have prophase, then we are going to, or prophase 1, then we're going to get into, so remember it's PMAT. So then we're going to get into metaphase 1. So metaphase 1 is next. So metaphase 1, middle, we have our chromosomes lining up in the middle. So it's actually our homologous chromosomes. So I'm going to show that here. We're going to have our centrioles on opposite sides. Now, We could have our chromosomes line up just like this. So maroon on the left and yellow on the right, pink on the left and blue on the right, or they could be flip-flop. So it could be purple, yellow, blue, pink, right? So that leads to more genetic variation on how they line up on the plate, the middle plate. So we'll show this one like it looks above, but then we'll flip-flop it for the other one. And again, this just leads to, depending on how it lines up, that's called independent assortment. It's independent of one another. So just because the purple one's on the left doesn't mean that, okay, the pink one has to be on the left. These homologous chromosomes are independent to these homologous chromosomes. And so it's just by random chance on how they line up in the middle. So we have our centrioles, I don't know why I do an arrow, connecting to our homologous chromosomes. So metaphase 1 looks different than mitosis metaphase. Remember, in mitosis, the chromosomes line up just straight down the middle, like that. So they're not in their homologous pairs. Where in meiosis metaphase 1, the homologous pairs are lined up in the middle. And so that's pretty important. So that's how you would state that, the homologous pairs, or you could say the tetrad line up in the middle of the cell. Okay, after metaphase, then we get into our anaphase 2. Or sorry, anaphase 1, my bad. So anaphase, again, just like before, we have that happening. Do we have pink on the right side? Cool. And so in anaphase, we have our spindle fibers are pulling our chromosomes, our homologous chromosomes apart. So homologous chromosomes are being separated from each other. And again, remember, we're just showing 4 in this example. In reality, we have a total of 46 chromosomes, so 23 pairs of chromosomes are going to be separating from each other in human cells to form the gametes. Cool. So then after anaphase, we have telophase 1. Make our cell into that little peanut shape. Our nuclear envelope starts to form around the 2 clusters of chromosomes. So because I have the maroon and the mostly blue on the left side, that means that those 2 have been pulled to the left side of the cell. So I need to show those ones over here. Right, so the homologous chromosomes should end up in opposite cells. Okay, cool. So we also have cytokinesis that happens. So if I were to say like telophase / cytokinesis, so these would end up breaking apart from each other. Ah, snap. There we go. And so now we end up with 2 cells, but that is the end of meiosis 1. And then we go straight into meiosis 2. So meiosis 2, we're starting off with these same cells. So we're just going to be watching these same cells. So we go into prophase. Prophase 2. So prophase 2 is going to be basically those 4 steps that we talked about, not including the homologous chromosome crossing over, because the homologous chromosomes are now in opposite cells, right? So we have purple and yellow together. Well, in our new cell, the purple is in one cell and the yellow is in the other cell. So there's no way that they can pair up because they're not even in the same cell. So we just have our 4 things that happen in mitosis that are happening in prophase 2. Our centrioles are separating, our spindle fibers are starting to form, our nuclear envelope breaks down and our DNA coils back up into that chromosome state. Let me draw those really quickly. Okay, sweet. So those 4 things happen in prophase 2. Again, those are the same things that are happening in mitosis. So after prophase 2 of meiosis 2, then we get into metaphase 2. Metaphase 2, the chromosomes line up in the middle of the cell. So basically kind of like how it looks. Now this, technically these don't have to already be in the middle, I just drew them like that. But this spindle fibers, so I'm just going to use the same picture that's up here. The spindle fibers are attaching to our centromere and they're helping line these chromosomes up in the middle. Now, they could, again, line up where we have like mostly yellow and a little bit of maroon and then mostly pink and a little bit of blue, but that could be flip-flopped. So we could have the little bit of blue on the other side, right? Just because the little bit of purple's there, we could have it on this side, which again leads to more genetic variation. And then same thing over here. You could have had that little bit of yellow on that side or the little bit of pink on that side. But we'll just draw it this way and see how it ends up looking in the end. So that's metaphase 2. After metaphase 2, then we get into anaphase 2. Anaphase 2 looks very similar to mitosis anaphase. So again, our sister chromatids. So in anaphase 2, it's the sister chromatids separating. Or in anaphase 1, oops, was that at the top of this one? It's the homologous chromosomes separating. So anaphase 1 is the homologous chromosome separating, and in anaphase 2, it's our sister chromatids separating. Cool. And then we have the blue one. The pink. Cool. All right, so then we get into telophase. /cytokinesis. So again, telophase has that like peanut shape. And our nuclear envelopes begin to form around the 2 clusters. And well, I guess we have, so see how this purple one is going to the left? So I'll show that there, mostly purple, a little bit of purple. I guess it's maroon, I don't know why I keep calling it purple. And then our yellow, so that one has a little bit of yellow on it. This one is yellow, and that one's fully yellow. Cool. And then we have our other 2. So our blue, mostly blue, a little bit of blue with the pink. And I know that we've gone through this multiple times before break, but I know it's been a minute since break. So then telophase, remember that the cytoplasm completely pinches off. So let's see if I can do that without getting wild. Now will it delete the whole thing again? Oh, yeah, I'm sorry. So there's 2 cells. And there's our other 2 cells. So we have a total of 4 cells. 4 cells. 1, 2, 3, 4, right? And each of them are genetically different. This one has mostly purple with blue. This one has a combo of mostly blue with a little bit of pink, mostly maroon with a little bit of yellow. This one has mostly yellow with a little bit of maroon, all pink, mostly pink with a little bit of blue, and all yellow. So again, all 4 of these cells are genetically different from one another. So if these were in males, then these would all turn into sperm. And if this was the sperm that ended up fertilizing the egg, then that zygote or baby would end up having these genetic combinations. Where if it was this one, then it would have slightly different genetic combinations. Where if you were looking at this combination versus this combination, they are completely different from each other, right? So the genetic combination, just depending on even within one individual, which sperm ends up fertilizing the egg, you can have a lot of variation. And that's why you and your siblings aren't identical to each other, because you have this crossing over and how it lines up on the metabolic plate. It changes the genetic variation, which is pretty cool. Okay, cool. So that's our little review of mitosis. So if you have questions, let me know. Send me a remind text or an email, and I can help you guys out. But hopefully that was a good review and you guys feel ready for the worksheet. And again, your quiz is tomorrow. After the quiz, we will start on protein synthesis. So that's the process of making protein. So remember that our chromosomes, so each of these little guys that we just made down here, these genes or these chromosomes have genes on them, and the genes are coding for specific proteins. So we're going to say, oh, if we are specifically looking, let me zoom back in again. Let's say we're specifically looking at like this area of that specific DNA, what protein would we be coding for? So how would that work? Which is kind of cool to get into. Okay, sweet. So if you have questions again, shoot me an email. I will talk to you guys later.